Good morning, everybody. Thanks for joining in. You guys are right on time, unlike me, so I'm a little late. We still got a minute, actually. Rayna, thanks for presenting first. I wanted to remind people about the proposal, and there have been, or the assignment, people have been asking about the length. And I think what I'm going to do, or I am going to do, is say you guys can use 2 pages just for the research and then additional pages for references and figures because people are already doing that, handing it in, and I think it's just simpler if everybody just has the same 2 pages for your story, and you're not judged. Like, it doesn't have to be really dense. It shouldn't be. It should be a good story. And then you can add an additional page for references and let's say 2 additional pages for figures. Does anyone have any questions about that? I'll put it up on the e-class as well so you can see that. But Rayna, why don't you share your slides, and thank you for going first, and also just unmute.
Sounds good. Can everyone hear me?
Yeah, that sounds great. Okay, that looks great. Just coming up. And thank you. You can start in a second. It's just about to come up, I suspect. Okay, great. Go for it.
Awesome. Good morning, everyone. Today I will be talking about a member of the dark kinome called P21 activated kinase. It's also known as PAX6. So the PAX6 is a serine-3 kinase, and it's a member of the sterile 20 family of the STE group of serine-3 kinases. PAX6 uses ATP and transfers a phosphate group from ATP onto serine or threonine and maintains a basal activity. Most notably, PAX6 interacts with the androgen receptor by binding to the ligand binding domain of the androgen receptor and phosphorylating its DNA binding domain. This inhibits AR-mediated transcriptional activities. When PAX6 phosphorylates serine 50 or 578 on the androgen receptor, binding of the receptor to MDM2 is enhanced, which leads to further phosphorylation and ubiquitination, leading to AR degradation. While PAX6 is not ubiquitously expressed in all tissues, there are many tissues that it is expressed in. The GTEx results show that PAX6 is primarily expressed in brain, skin, testes, esophagus, mucosa, and the vagina in comparison to other tissues. Experimentally, using northern blots, research has shown that PAX6 expresses primarily in the brain, testes, breast, and prostate. PAX6 is generally localized to the cytoplasm, but when it is interacting with the androgen receptor, it translocates with the receptor into the nucleus, where PAX6 will inhibit the androgen receptor. PAX6 has 2 main domains. It has a kinase domain, which is in green, located by the C-terminus, residues 407 to 658, and a crib domain in pink, which is located by the N-terminus with residues 12 through 25. The region between these 2 domains is a linker domain. The protein is predicted to be globular and cytosolic, and there's no evidence of any coiled coils or transmembrane sections. Disoliprid demonstrates that there's potential for lipid binding at the N-terminus and C-terminus of PAX6, as well as approximately halfway through the protein in the disordered region. This is confirmed with MODA, where potential lipid binding occurs in the linker region primarily and at the N-terminus. AIUPRED predicts that there's disordered protein regions in the linker region at the N-terminus and close to the C-terminus. These regions are likely flexible and in order to help with the interaction between PAX6 and other proteins like the androgen receptor. In the PAC family, PAC1, 2, and 3 are all activated by small GTPases, and these GTPases interact with a crib domain, as can be seen here in this example of PAC6 crib domain with CDC42. In PAC6, the small GTPases don't seem to activate PAC6, but it may be involved in the translocation or localization of the protein. In terms of post-translational modifications, there's no lipid modifications or methylations. There's predicted to be one ubiquitination and many phosphorylations, primarily in that linker region, as seen on the lollipop plot monophosphorylation of serine 560 and phosphorylation of threonine 566 by MAP kinase 6 contributes to the activation of PAX6. And there are possible pip stops in the linker region due to the disordered membrane binding regions. There is a potential of PAX6 to be involved in cancer. From the dark kinase knowledge base, there's evidence of missense mutations and mRNA upregulation of PAX6 in cervical squamous cell carcinoma and endocervical adenocarcinoma. And there's amplification of PAX6 and upregulation in prostate cancer, which plays a role in its malignancy. And this is seen in research that's been done. In the literature, research shows that activated PAX6 by androgen stimulation and subsequent interaction with the androgen receptor leads to promoted prostate cancer cell motility and invasion. PAX6 inhibition and irradiation results in significantly decreased survival of prostate cancer cells, demonstrating that PAX6 is involved in prostate cancer promotion.
Great.
Here are my references.
There are going to be questions in the chat space. I'm going to ask you to answer them because we're a little short on time. Great talk, but you went a little bit over. So if it's okay, we're going to just ask you to respond and have the next presenter share their slides. Thanks, Rayna. That was great. So Riley, are you here? Perfect. And thanks for asking questions, Justin and Ty, and Rayna will answer those in the chat space. Riley, your screen is just coming up, and as soon as it appears, you're welcome to kick off. And sounds like you've unmuted, so that's great.
Okay, awesome. I wasn't sure how to do everything. Sorry. Can you see my screen? Okay.
I can. Just if you could full screen, that would be great.
Yes. All good.
Go ahead.
Okay. So my chosen protein was cyclin-dependent kinase 15, or CDK15 for short. This is Unipart entry I used, and this is enzyme class. So CDK15 is a serine threonine kinase, and like the rest of the CDKs, it's involved in proliferative and cell survival pathways. CDK15 has been shown to interact with both PAC4 and survivin. CDK15 phosphorylates PAC4 on its serine 291 residue, which activates it. It stabilizes it in its active form. PAC4 is involved in multiple different cellular pathways. It stimulates cell motility, drug resistance, resistance to apoptosis, and it also stimulates several proliferation pathways such as ERK shown there. Survivin, when phosphorylated, inhibits the TRAIL apoptotic pathway. TRAIL is an acronym, which is short for tumor necrosis factor related apoptosis inducing ligand. It basically just induces apoptosis, and survivin in some way, we're not quite sure how, inhibits that apoptotic pathway. CDK15 is primarily expressed in the ovaries and fibroblast cells, and it's predicted to be localized in the cytosol nucleus, but we're not sure. There's very, very little structural information available for CDK15, unfortunately. Just about everything's predicted. So that there is a alpha fold structure as seen in PyMol. I colored the serine threonine kinase domain blue, and it spans amino acids 103 to 387. There's two predicted sorter domains, one by the N terminus at the top left there, and one by the C terminus in the bottom right there, which I've colored in green. And in those disordered regions are multiple predicted membrane binding regions, which I've colored in red. Unfortunately, there were no signal peptides predicted by Phobius, and as expected, there's no transmembrane regions because it's predicted to be a globular protein. There's also a predicted coiled coil at the bottom of the active site in blue there. There's very little information available for the regulation of CDK15. We don't know what cyclin or what other protein binds to it. I did find 4 predicted pip stops though for CDK15. Serine, located at serine 49, threonine 36, cysteine 15, and serine 77. The first 3 pip stops are located between those first 2 red membrane domains at the top there. And then the serine 77 pip stop is located just in the bottom membrane binding region in red at the bottom there. It also has a predicted ubiquitination site in the active site at lysine 239. Unfortunately, there were no predicted prenylations and reliculations, but there are predicted palmitoylations in both the disordered N-terminal region and C-terminal region. So CDK15 has been mostly studied in the context of colorectal cancer and breast cancer. If you type in CDK15 in the University of Alberta's library search engine or in Google Scholar, that's pretty much all that'll come up. Despite that, though, it hasn't been shown to be involved in very many cancers, the most being 5% and uterine carcinomas. One research paper I found showed that it was overexpressed in colorectal cancer, and that overexpression then increased the expression of PAC4 because it was being hyperactive, and then that PAC4 expression increased metastasis, proliferation, resistance to apoptosis, et cetera, et cetera. However, it's also been shown to be downregulated in some cancers like breast cancer. One group found that a protein called PA28 alpha, which was an oncogene that's a part of a proteasome complex, actually inhibited CDK15. So when they inhibited PA28 alpha, the CDK15 levels suddenly spiked. Why or how CDK15 is inhibited in breast cancer is unknown, but it's shown to be inhibited despite being an oncogene. And yeah.
Great, Riley. Thanks so much. I think just in the interest of time, I'm going to keep rolling on and ask you to respond to questions in the
I
chat.
will, sorry.
But that was great. And let me just ask the next person to share their screen. And thanks, Riley, for answering those questions. I think Shadman is next.
Hi, yes, I can't share the screen for some reason. I think like there's a issue
Okay.
with like the permission.
Yeah, you can do it now. I just, I think active.
Yeah, let me just try.
Perfect.
Can you see my screen?
I can see your Zoom screen, but not your presentation. Ah, perfect.
Yeah, okay. Just give me 1 second. Yeah.
Great.
Is it okay?
You're good to go. Perfect.
Perfect. Thank you so much. So yeah, hi guys, I'm Shadman Sakib Khan, and today I'll be speaking about the dark kinase STK32A. So the dark kinase STK32A is a dual specificity AGC kinase with preferences for acidic substrates. The AGC group is basically named after 3 kinds of protein kinase, which is protein kinase A, G, and C families. And these serine protein kinases are basically regulated by secondary messengers, such as cyclic AMP for pKa and lipids for pKc. One funny thing is the naming is quite creative from the, I guess that's what you get when you get biologists to name certain stuff. So yet another novel kinase. And here's also the chemical reaction shown here. Yeah, so there are 3 known isoforms for STK32A and uniprot PDB. I've taken the canonical sequence here. So there has been only one significant domain, that's the catalytic or the kinase domain, that spans from 23rd to 281st amino acids, and then there's a disordered region which is from 373 to 396th amino acid. So here's also an AlphaFold structure, which basically shows the whole kinase. The dark blue indicates the regions with the highest confidence, and orange indicates the region with the lowest confidence. And then this is the expression pattern from GTEx. So this heat map basically displays the transcript per million values across various human tissues. The highest expression is observed in brain regions, particularly in the cerebellum, basal ganglia, and cortex, which suggests a potential neurological function. Moderate expression appears in the heart, kidney, thyroid, skin, ovary, and then low levels are detected in white blood cells, muscles, and other peripheral tissues. So for the cellular location, I think it's mostly bound to, it's basically bound to the cell membrane. It is most likely to be a peripheral protein. However, there's some conflicting information. Some sources say it's integral protein. But from Phobius and deep TMHMMM, so from the Phobius graph, you can see that there's a low probability of of being a transmembrane domain present in this kinase. However, given given this plot has cytoplasmic, non-cytoplasmic regions, some portions may be near domains, but they are not interacting. And also, since this is a globular domain, it basically suggests that it cannot be a transmembrane protein. And since there's no membrane spanning regions, it cannot be an integral membrane protein, so it has to be a peripheral protein. So here's the MODA structure. From the MODA output, we can basically see that the latter, or like the last portion of the protein, specifically the disordered region, has higher MODA scores. And yeah. And here's a GIF. I don't know if you guys are able to see it for the OPM output. Where blue is the inner leaflet of the plasma membrane, red is the outer leaflet. So for the biological function, it basically affects planar polarity. Planar polarity is basically an organizational feature of epithelia in which the polarity of cells and tissues is coordinated along a plane perpendicular to the apical axis, to the apical basal cell polarity axis. The STK328 plays a crucial role in regulating planar polarity in the vestibular maculae of the mouse inner ear. It basically divides it into two different parts, and then the GPR 156 through regulating GPR 156, it basically controls that duality, or like that division. And here are some post-translational modifications. There have been 3 phosphorylation sites, T175S194 and was 342, and then 1 methylation site in R195. There have been some mutations. So missense mutation obviously has the highest percentage here, and then there's some insertions and deletions. Mutation also include mutation included in increase in gene expressional that changes the expression of the gene. Primarily occurs in adrenal gland, also in esophagus and ovary. So for the diseases, I think the mutation is highly I'm sorry. Approximately 6% of the cases exhibit STK328 observations for the renal clear cell carcinoma. And then for the melanoma, sarcoma, and endometrial, around 3% cases support that. And then since this is a specific kinase with dual specificity, it could be targeted by specific inhibitors. And also, since it plays a small part in non-small cell lung cancer's growth, in preventing it will probably prove to be a great thing for developing drugs related to those mutations. And here are my references.
Great job. That was a great story. Thanks again. I wanted just to keep rolling because I think we're a little tight on time. You guys have got great stories, but you're going a little bit long, and so I want to keep on schedule because I know you're busy as well. So, Takdeer, if you can share your screen, that would be great. Okay, it's coming up, and unmute, and you're good to go.
Does everything look? Oh, I should press slideshow. Sorry.
That'd be great. Thanks.
Okay, there you go.
We can. Oh, perfect. Yeah, great.
It's good. Okay, sounds good. So today I'll be talking about cyclin-dependent kinase 18. So there are 2 primary isoforms for this kinase. We got an isoform that is expressed mainly in the brain, but it's also expressed in other tissues as well. And then we also got some expression in the spinal cord and the basal nuclei. So there are many different functions that I can't explain all of them in this 4-minute little presentation that I have, but the main function that is evident for this kinase is oligodendrocyte differentiation. We also got some cell cycle regulation from G1 to S phase, and some cyclin-dependent protein serine threonine kinase activity. And then we also see some apoptosis regulation via P53 with this kinase. So regulation is primarily driven by this DFG motif right here. So this regulates the magnesium, which helps with ATP binding site at these residues. And there's two primary ways of activation of this kinase. So there's one way of phosphorylation by pKa at these residues, and then there's interaction with cyclin 2A. So here's a here's like the lollipop diagram from the phospho site. We see some key phosphorylation sites at these residues. However, these didn't really correspond to the literature that I was going through. So there is some room for speculation. But in terms of the literature, I think I'm gonna rely more on the literature just because there are some experimental findings in that regards. So with structure, with Swiss palm, there's no myristoylation present. There's no palmitoylation, farnesylation, nor prenylations. There's a serine threonine kinase domain from 144 to 425. So here is a PIMO diagram for CDK18. We got a lot of alpha helices, and this can be confirmed through the deep coil database. We see a lot of peaks. In MODA, we see a bunch of peaks here, but I think these are false positives because throughout my research, I've seen that this kinase primarily either cytoplasmic or located in the nucleus due to the cell cycle regulation part of things. So here on the left, we can see that this kinase is primarily in the inside, which makes sense. Phobius, I was a little bit skeptical with just because there's some non-cytoplasmic region, and I think Phobius doesn't detect nuclear localization signaling, so I think that's probably why. The cytoplasmic aspect can be correct with some functions, but there's a lot of functions that are regulated within the nucleus. So I think it's just that part that Phobius didn't catch. So there are many diseases that are linked to CDK18. I will talk about the oligodendrocyte differentiation part of things. So I made this little diagram to help connect everything from the literature. So when we get some deep deletion of the 2 copies of the gene, we get low CDK18 expression. So this can result into low ATK kinase signaling. And ATR kinase signaling, there's 2 main components that the CDK interacts with which is RAD17 and 9, and these are checkpoints that help with DNA integrity. So when we don't have enough CDK18, this will not regulate the ATR kinase signaling, thus will result into DNA damage as this cell cycle is stuck at this S phase. This will result into low differentiation of oligodendrocytes, which can result into demyelinating disease. So demyelinating disease is damage to nerve cell protection layer, the myelin sheath, and this can result into multiple sclerosis. And so because of this damage to neurons, this can also result into neurological diseases. So there is a bunch of research on how high CDK18 can also impact disease. So we've got some other diseases here, such as Alzheimer's, cancer, and depression, but I believe there's still much research to be done in these areas. And yeah, here are my references. Any questions?
Okay, great job. I think I'm going to ask you to just respond to questions in the chat space, if that's okay. And but it was great, great slides again. And then I'm going to ask Taylor to share her screen. Thanks, Taylor. And
Okay.
it's coming up. That's great.
Can you see it okay?
It's just appearing in 1 second, I suspect. Okay, great, perfect. Ready to go.
Okay, perfect. Okay, so the protein I've chosen is NEC11, and it's also known as NEMA-related protein kinase 11. And then there's the UniProt entry and the enzyme class there. And this is just the PIMOL structure of that protein. NEC11 is a serine threonine kinase with its active site residue at aspartate 158, which is on another slide highlighted. It is involved in regulating the cell cycle when there is DNA damage, specifically ionizing radiation damage to DNA. And it will stop the cell cycle at either the G2 mitosis checkpoint or at the G1S checkpoint. And the pathway on the left here is stopping it at the G2M checkpoint, where there is DNA damage, which will then activate ATM or ATR, activating CHK1, which can phosphorylate NEC11 at serine 273, and primes CDC25A, so that NAC11 can then phosphorylate that at serine 82, targeting it for degradation and stopping the cell cycle at the G2M checkpoint, as CDC25A can't activate CDK1. For regulating the cell at the G1S checkpoint, it involves phosphorylation by NEC2A, which will then allow the two to form a complex together. And the total pathway there hasn't fully been recognized or figured out, so I don't have a pathway for that yet. For regulation, there is autoinhibition of the C-terminal non-catalytic region, inhibiting the N-terminal catalytic region through intramolecular binding, which is shown on the right. It's not bound in this this structure, obviously, but the pink shows the C-terminal region and the orange is the N-terminal. And phosphorylation at serine 273, which is right here, by CHK1 activates the protein to stop it at the G2M checkpoint, and phosphorylation of the C-terminal serine residue by NEC2A will also activate the protein to stop it at the G1S checkpoint. For expression and localization, it has broad expression, and there are 4 isoforms, the main isoform being NEC11L, is commonly found in the testis, thyroid, and 22 other tissues, which can be seen on this graph here, and it is found in the nucleus and nucleolus. For structure and domain, Phoebus shows that it is mostly a non-cytoplasmic protein with a slight transmembrane. However, it's not very high probability, so it is likely just not very significant. The motograph does show a very significant spike around residues 360 to 380, which lines up with the coiled coil domain, which is in the same region. So I believe that the spike in the motograph is likely just due to the coiled coil region and not actually a membrane interacting region, as it is predicted to be a globular protein, so it wouldn't be interacting with the membrane there. Disolipid shows that there are quite a few lipid binding regions, which I believe to be involved in protein-protein interactions, as there is one on the N and C terminal, which are known to bind for autoinhibition of the protein. And it forms a complex with NEC2A, so it is likely that one of the other sites is involved in the formation of the complex with NEC2A. There are no lipidations, and for phosphorylations, the key phosphorylations that I was able to determine were serine 273, which is activation by CHK1, and serine 387 is likely the site of the phosphorylation by NEC2A for that regulation there. There is one acetylation site and there is one ubiquitination site as well. As for disease-linked mutations, loss of the function could lead to susceptibility of melanoma. However, it has not been fully identified whether it is the cause of it. Most of the mutations are missense mutations that don't seem to have any pathogenic effect, which can be seen in the UniProt diagram on the bottom here. And it was involved in a study involving somatic mutations in kinases linking to many types of cancers. However, it was not one of the main kinases studied. It was just one of the ones possibly involved. So it is likely not a cause for any specific types of cancer. And the bottom diagram here just shows the percent samples with mutation, with the highest percent being 4% missense mutation in the uterine corpus endometrial carcinoma tissue. And overall, it has been linked to increased incidence of colorectal or ovarian cancer, but has not been the direct cause of any specific cancers or diseases. And here are my references, and are there any questions? I'm not sure if the audio cut out or anything, but I can't really hear anybody just in case something happened. So I'm just going to answer questions in the chat.
Sorry about that, Taylor. I had a little computer issue there. Can you hear me, Taylor?
Yep.
Okay, that was a great presentation. Apologies for the technical difficulty at this end, but I'm going to ask you to answer chat questions, and then maybe we can move on to Teryn, if that's okay. Great. Your slides are coming up,
Okay,
and
perfect.
we can see you and hear you. Perfect.
Okay, hi, I'm Teryn, and today I'm presenting on testis-specific serine threonine kinase 1B, or TSSK1B. Here's its predicted structure from AlphaFold. Testis-specific kinase 1B is required in spermatogenesis for cytoplasm reconstruction during sperm tail maturation and for the formation of the annulus at the flagellum base. It phosphorylates testis-specific kinase substrate by binding ATP and also magnesium ions, probably to stabilize ATP and facilitate the transfer of the phosphate group from ATP to TSKS. TSSK1B is expressed in sperm during late spermatogenesis and functions in the cytoplasm, acrosome, flagellum, nuclear bodies, centrosome, annulus, and the perinuclear theca, which surrounds the nucleus everywhere but the tail region. Here are some photos from UniPro and the Human Protein Atlas that highlight the expression, here in beige and here in green. And then here, as you can see from SwissPalm, the protein is entirely cytosolic. Structure-wise, TSSK1B is globular. Here's a structure from Pimal. I've colored with some of the important domains and binding sites. You can see the kinase domain in pink, the ATP binding site in blue, and the kinase active site in red. Another important site is aspartate 136, which is the active site proton acceptor. The disordered region shown here from IUPRED3 could be important for protein-protein interactions with proteins like TSSK2 or heat shock protein 90. Heat shock protein 90 is important for stabilizing TSSK1B by negatively regulating ubiquitination. And then here, Phobius shows a transmembrane domain, but it's in the kinase domain and correlates to an alpha helix that's embedded inside the protein in this area. So this is likely a false positive due to hydrophobic residues. TSSK1B is activated by phosphorylation of threonine 174. A paper I looked at said TSSK1B can autophosphorylate threonine residues. So this activation could potentially be done by autophosphorylation. Here's the protein with T174 colored in blue and the active site in red for reference. And then here are the results from phosphocyte. The ATP binding region spans from residues 18 to 41. So these phosphorylations at residues 22, 23, or 31 could be involved with regulation of ATP binding. So on to complications with this kinase, threonine 174, which is that residue that activates TSSK1B when phosphorylated, can be mutated to an alanine, which would make the kinase permanently inactive, which could potentially cause male infertility since TSSK1B is involved in the last steps of spermatogenesis for sperm function, although TSSK2 could compensate since they have high shared identities. So this is a little inconclusive. If that T174 is mutated to a glutamate, the negative charge would mimic phosphorylation, so the kinase would always be active, but there's not much information on the implications for this. Here's a chart from the Dark Kinome website that shows TSSK1B mutations in cancer. By far, the most prevalent cancer is colon adenocarcinoma, which is interesting because TSSK1B is only expressed in the testes according to UniPro and the Human Protein Atlas. QPTM also yielded some interesting results since there was 9 different entries that showed phosphorylation of serine 156 on TSSK1B in ovarian and breast cancers. So there's definitely some additional research needed here. And that's all I have today. Do you guys have any questions? Dr. Overdoon, you're muted.
Okay, sorry. That was great. You covered a lot of important information there. Thank you for sharing your story. And I'm going to ask you to respond to questions in the chat space. You did a great job, though, again. They've all been great talks, in fact, today. Tiffany, are you able to share your screen? And yeah, we're just a little behind schedule. If people need to leave, that's okay, I understand. But if you can hang out and ask questions, that's great as well. Thanks. We can see your screen, and if you can full screen, perfect. That looks great, Tiffany. Go for it.
Can everyone hear me?
Yep.
Awesome. So, hi everyone. The kinase I chose to research is serine threonine kinase 40, also known as STK40. STK40 is predicted to negatively regulate NF-kappa B and P53 mediated gene transcription, NF-kappa B, also known as nuclear factor kappa B, and P53 are transcription factors that drive pro-inflammatory and tumor suppressor gene expression, respectively. STK40's exact mechanism is still under study, but it may act as a scaffold via COP1 or constitutive photomorphogenesis 1. So as shown in this diagram, COP1, an E3 ubiquitin ligase, binds STK40, triggering ubiquitination and degradation of key signaling proteins such as P65 in the NF-kappa B pathway and P53 in its own pathway. STK40 is broadly expressed in humans with strong expression in the heart, brain, placenta, etc. It is localized to the cytoplasm and nucleus, which is supported by data from signal P. So this graph over here shows that there are no signal peptide sequences, so it is not targeted for secretion. So there are 4 key features of this kinase. First, it contains a kinase-like domain made of residues 35 to 332, shown in figure A here, highlighted in yellow. Substitutions in conserved residues prevent ATP binding, making it a pseudokinase. The particular substitution that prevents ATP binding is alanine to glutamine at residue 64 because glutamine projects into the site that ATP would normally bind at. Second, figure B highlights 2 ATP binding sites highlighted in lime green, which are residues 41 to 49 and 66. However, ATP doesn't actually bind to these regions. Lastly, the proton acceptor site highlighted in forest green at residue 196 may contribute to STK40's scaffolding function. The third thing to note are the membrane binding sites, which I will go into more detail in the next slide. So in figure C, I circled the locations of these membrane binding sites, and as you can see, there are 4, all of which may transiently interact with membrane sites through COP1. Finally, Phobius data in figure D shows that there's a transmembrane helices from residues 257 to 275. Given that this region is within the kinase domain and positioned internally, which is hydrophobic like a transmembrane domain, I concluded that this data is a false positive. The protein may open up to expose this region, but that is very unlikely. So going back to membrane binding sites, deep coil analysis shown in figure A shows no coiled coil regions, indicating that it doesn't multimerize. Figure B presents MODA data, which I graphed, and it highlights that most membrane binding sequences cluster at the C terminal. So a study by Derzinka and her research team determined that STK40 has a potential binding motif for COP1 at the C terminal, and this aligns with the proposed mechanism of STK40. Figure C from disoliprid further supports this, predicting 4 lipid binding regions, primarily at the C terminal. Moving on to STK40's regulation, phosphosite predicts multiple MET and PIP stops as shown in the lollipop diagram. However, only 3 of these are significant because most post-translational modifications are outside the membrane interacting regions. Phosphorylation sites at residue 406 and 422 suggest that they regulate C-terminal interactions with membranes, and a MET stop at residue 30 supports potential N-terminal membrane interactions. So in this PyMol structure that I made here, I highlighted the PIP stops in red and the MET stop in orange. STK40 is primarily studied in cancer due to its potential role in negatively regulating tumor suppression genes like P53. This is why STK40 is predicted to be overexpressed in many cancers, suggesting an oncogenic role. According to Cosmic, the most common mutation is C to T, making up almost 41% of all mutations. It is a missense mutation mainly linked to carcinomas. Other disease-related mutations remain poorly documented and require further research. And this pie chart that I made illustrates STK40 mutation distribution across tissues, with highest occurrence in female organs like the vulva, endometrium, and placenta. And these are my resources.
Great. Thank you so much. That was really clear. Thanks so much for yet another great talk. And maybe you can answer those questions in the chat space. And we're getting close to back on time. Thanks for being on time, Tiffany. Ty, can you share your screen?
Yeah, hi, can you hear me?
Yes, perfect.
I have to share my screen here.
Great. It's just coming up. And just full screen and you're good to go.
There, can you see that?
Perfect.
Okay, so for this project, I chose CDK13. CDK stands for cyclin-dependent kinase, and cyclins are proteins that bind to CDKs, causing a conformational change, making them more active. It's part of a serine-threonine kinase family, and we know that it's localized to the nucleus. CDK13 is unique to other CDKs because it phosphorylates RNA polymerase II and moderates splicing factors, which are both major role players in the cell cycle. So CDK13 is important because it also has a major role in regulating the cell cycle, and it's been shown recently to be involved in different types of cancers, making it a potential target for drug therapeutics in the future. So just a bit about the overarching pathway. So it's not part of a signaling pathway, so to speak, but it's actually rather always present in the cell, in a functioning cell. So as mentioned before, it needs to bind to its specific cyclin, cyclin K, to cause a conformational change, allowing it to be more active. It also needs to be phosphorylated for further activation by its specific kinase, cyclin or CDK activating kinase 1. And then once it's in its phosphorylated and cyclin-bound state, it can phosphorylate RNA polymerase II, which is its target, which will increase the processivity of RNA polymerase II. It can also directly phosphorylate splicing factors, which will change things like the expression, localization, and activity, things like that for these splicing factors. So some predictions. So phosphocyte predicts a phosphorylation here in the kinase domain, which is good because we know that it needs to be phosphorylated. But Phobius predicts 2 transmembrane helices, which is more than likely false positives because we know that it's localized to the nucleus and not embedded in the membrane. And so MODA predicts 2 membrane interactions, both one at the N terminus and one at the C terminus, which is also more than likely false positives because these are just disordered regions of the protein, so it's not plausible. And then this is the disolipid graph. It predicts 2 membrane interaction regions, one on either side of the kinase, which is interesting because this could cause like docking to the membrane while not in use. But the literature doesn't show much on this, so it's a future area of research potentially. So since the structure is solved, just a little bit about structural information. So this is CDK in green here, and then cyclin K in purple with an inhibitor bound at the active site taken from the PDB. So this marks the N terminus and the RS domain. So the RS domain would be here, and it's rich in arginine serine, which is important in binding RNA polymerase II, its target. It's also important in binding nuclear import machinery, so it can get across the nuclear envelope. This is the C terminus marking the C terminal domain here. And then this is the active site in T loop, so active site here, T loop in here. And without cyclin bound, the T loop would cover the active site, binding to a catalytic aspartate in the active site via salt bridge, which would hold it inactive. When cyclin binds, it causes that conformational shift, which allows the T loop to back off, that bond will break, and then the active site will open up. And then this is the Piche helix, which is common to other CDKs and responsible for binding cyclins. And then I just wanted to highlight the phosphorylation here at this site for further activation. CDK13 is an emerging area of research. It's not all the way uncovered yet, but we know that it plays a crucial role in neuronal development and neuron proliferation, as well as it's been shown to be involved in certain types of brain tumors. Inhibition of this CDK in certain types of brain tumors increases the efficacy of certain treatments. And then here's just an image of the inhibitor bound at the active site. Any questions?
Great job. Thank you so much. And I think we're just going to try and keep on rolling and ask you to answer questions in the chat space if that's okay, Ty, but that was very well presented. Zach, can you share your screen and unmute as well? I want to thank you all for great presentations and answering questions in the chat space today. Apologies, it was a pretty packed schedule, so we were just needing to kind of catch up on time a little bit. But it's great that you're answering questions in the chat space, and feel free to continue. If you can hang out a little longer, Zach, maybe you can unmute as well.
Yeah, sorry, I'm just have to give it access to share my screen, and it's making me quit, so I'll be right back. Sorry.
Okay, that's fine. I'll hang out. You should be able to share your screen. Is that not working for you? Okay, I think Zach is going to step back in a minute. Are there any questions for me? You've all done a great job, and I think hopefully it's prepared you for completing your assignments as well. Just as I mentioned at the beginning, you do have 2 pages of text that you can write, plus an additional page of references. You don't need to fill it all up, of course. Like a dozen references might be typical. And then also, and you don't need to refer to the papers for the tools. I mean, you can if you wish, but I already referenced them in my talks, and so that's understood. And then also 2 pages for figures that you can refer to in the text as figure 1, 2, 3, whatever. All right, Zach, you're back in. Oh, great. We can see your screen and just full screen and unmute, and you're good to go. Yeah, it's under slideshow. Perfect. That looks great. Just unmute.
Okay, last presentation here. So I'm going to be talking about branch chain alpha keto acid dehydrogenase kinase, or BCKDK for short. So like the name implies, BCKDK is a kinase involved in the metabolism of branch chain amino acids. So that would be valine, leucine, and isoleucine. It phosphorylates the branch chain alpha keto acid dehydrogenase complex at serine 293 of the E1 alpha subunit of the complex, leading to inhibition of the enzyme. And since this is the rate limiting step of the pathway, it greatly reduces the catabolism of branch chain amino acids. The enzyme is expressed broadly across all tissues, although the lowest levels are seen in the brain and highest levels are seen in the whole blood. BCKDK is localized primarily in the mitochondrial matrix, although it can also be found in the cytosol of hepatocytes, where it actually plays an important role regulating the activity of ATP citrate lyase, which is an enzyme involved in lipogenesis and gluconeogenesis. So here I have the alpha-fold structure of BCKDK, and you can see I've colored the 2 domains red and green, while the mitochondrial transfer peptide is shown in blue. So the domain in green is the mitochondrial branch chain alpha keto acid dehydrogenase kinase domain, which is pretty much a 4-helix helical bundle. And the domain shown as red is the histidine-like ATPase domain, which consists of both alpha helices and beta sheets. So like I said, the protein is mainly localized to the matrix, and this is due to the presence of mitochondrial transfer peptide at the N-terminus of the protein. So on the bottom of the slide, I've shown the target P predicts the presence of this mitochondrial transfer peptide, and that it will be cleaved off after arginine 30. So we can look at another structure here, which is actually a crystal structure obtained of the protein, and here the mitochondrial transfer peptide has been cleaved. This also shows the interaction of the protein with one of its substrates, which is ATP. So ATP is shown in blue, and then the interaction is mediated by a potassium ion and a magnesium ion. So those are shown in purple and green. And then the residues involved in ATP binding, I have highlighted in orange. So in terms of regulation, on the top right figure, you can see that D-coil predicts that the protein has a coiled coil motif in the BCDHK domain. If the prediction is accurate, it could be that this is involved in interaction between BCKDK and the complex. On the top left are the results from MODA, which predicts a couple of very short membrane binding regions, primarily also in the BCDHK domain. On the bottom left is the AIU pred prediction, which shows that there are no disordered regions. And finally, we can look results on the bottom right from Phosphocyte Plus, which shows a number of post-translational modifications, including phosphorylations, acetylations, and ubiquitinations. The majority of these modifications are also in the BCDHK domain, so they could be mediating that interaction between my kinase and the complex. But there are no PIP or MET stops because none of these are close to the membrane binding regions predicted by MODA. So last, I'll talk a little bit about the biomedical significance of BCKDK. So maple syrup urine disease or MSUD, is a metabolic condition that results in high levels of branch chain amino acids in the bloodstream and characteristically sweet-smelling urine. The disease is caused by mutations that decrease the activity of the branch chain alpha keto acid dehydrogenase complex, and the severity of the symptoms can vary from life-threatening to very mild and intermittent. So the gain of functions mutations in BCKDK can impair BCAA catabolism, but the phenotypes for maple syrup urine disease caused by that are usually quite mild and with like intermittent symptoms. And then BCKDK is also connected to diabetes, both through its direct effects, where it phosphorylates and activates ATP citrate lyase in hepatocytes, and by increasing levels of branched chain amino acids in the plasma, which can lead to insulin resistance. The treatment of obese rats with an allosteric BCKDK inhibitor has actually been shown to decrease hepatic triglyceride levels and improve glucose tolerance and insulin sensitivity. It is also associated with cancer. So BCKDK has been found to promote tumorigenesis of colorectal cancer by directly phosphorylating serine 221 of MEK, which is a downstream kinase of the EGFR signaling pathway and is involved in cell growth and proliferation. BCKDK is frequently overexpressed or mutated in both colorectal and other forms of cancer. And this could be important for diagnosis since BCKDK has potential to serve as a clinical biomarker. And it could be important for treatment because there are a number of known inhibitors of BCKDK, which could be used to prevent tumor growth. Does anyone have any questions?
That was great, Zach, very clear. Maybe you've got a second or two before 10:50. Ty asks, is the inhibitor for this kinase marketed yet? There's a couple questions in the chat space. I don't know if you can see those, Zach. Maybe you can respond in the chat space.
Yeah, so the inhibitors are not super efficient, so they have a lot of really bad side effects. So there's lots of work going on right now to try to figure out some more efficient inhibitors that could more specifically target the kinase without having any off-target effects. But it is like a pretty promising area of research going on right now.
Okay, there are a couple more questions in the chat space. Feel free to answer those either verbally or in the chat space. And thanks everyone for showing up and presenting great talks and all the questions and answers. I look forward to seeing you on Monday. We have Dr. Rajiv Nair speaking in the classroom, same room. It'll be, I think, our last class, actually. So thanks very much for coming on Monday at 10 o'clock, as usual. And really appreciate all of your hard work on the talks and the assignments. I'm going to set up also a Google form so that you can upload them. Email also works, but it's a bit easier for me if you can just upload them to the Google form that I will share on eClass. Thanks again, everybody. Happy to hang out and answer questions. If you want to unmute and ask me anything about whatever, feel free to unmute and I'll just stay here for a bit longer. I'm going to just turn off that recording.